welcome to today's video lecture. In today's video lecture, I'll be solving previous year's NEET questions and discussing the answers. This is for the chapter, as you can see on the screen, Human Health and Diseases, and we are in part 3 of the series. So if you haven't watched part 1 and 2, please do so. Alright, so let's begin. Alright, so question number 1. The infective stage of malarial parasite Plasmodium that enters human body is... This appeared in 1990, pretty old question. A. Merozoite, B. Sporozoite, C. Trophozoite, and D. Minuta form. Okay, so the infective stage, we've already discussed this a million times and it's very, very important. We need to learn the life cycle of Plasmodium. There is no way out. So please read NCERT. So you will get questions related to life cycle of Plasmodium. Alright, so the infective stage of malarial Plasmodium is Sporozoite. Alright, that's the infective stage. That's what's present in the salivary glands of the mosquito, which then goes into the human body. Okay, let's move on then. Yeah, so the infective stage of malarial parasite Plasmodium is sporozoites. Okay, we discussed this many times. Okay, so question number 2. The bite of CC fly may pass to humans. Okay, so whatever that pathogen is. This appeared in 1989, another very old question. A. Leishmania donovani, B. Trypanosoma gambians, C. Entamoeba histolytica, D. Plasmodium vivax. Alright, so CC fly is the vector which carries Trypanosoma gambians. Okay, that's called African sleeping sickness. Okay, so the answer is B. And I've discussed this in previous lecture also. You're familiar with this table, I'm sure. If you've watched part 2 of the series, you would know we already discussed this. So this should be very easy. So we have West African sleeping sickness, which is caused by Trypanosoma gambians, and East and Central African sleeping sickness by rhodocins. Then you have Central and South America. So it depends on which part, okay, which part of the world it is, or which part of the African continent are we in, okay, the West African or the East and the Central African, over there in Central and South America. Okay, we have Trypanosoma cruzi, which causes sleeping sickness. Okay, so and I've also mentioned the vector. So basically you will have you'll get questions related to this as we have seen. Remember this table, it'll be easier for you, right? So but a pretty direct question, right? If you go back, it was a pretty direct question. The bite of CC fly is may pass to humans, Trypanosoma gambians. Okay, so let's move on then. Alright, so question number 3. Malaria fever coincides with liberation of, this appeared in 1989, A. Cryptomerozoites, B. Metacryptomerozoites, C. Merozoites, and D. Trophozoites. Okay, so I haven't discussed this when we discussed the life cycle of Plasmodium, but it's nothing, it's simple. We know that merozoites, when they enter the erythrocytes, the breakage of erythrocytes result in fever, right? The malarial fever, which is the clinical form. But there are different names given to the merozoites. So in this case, malaria fever coincides with liberation of metacryptomerozoites. Okay, that's B. And we'll come to the discussion and explain it to you further. So the answer is B. Okay, so the merozoites that enter the bloodstream, and remember this, okay, and attack the erythrocytes are called metacryptomerozoites. Now, basically, based on the generation of merozoites, there are different names given to it. That's all. There's nothing complicated about this, but learn it as a fact. Okay, so it's actually second generation of merozoites. So the merozoites that enter the erythrocytes, and they are called metacryptomerozoites. Now, the first generation of merozoites that form from the sporozoites from the liver cells is called cryptozoites. Okay, so they are first generation. So the merozoites basically have been given names, okay, based on which generation they belong to. This cryptozoites is metacryptomerozoites. So based on that, that's all. So, but the life cycle obviously remains the same. There is nothing different about that. It's just that the Merozoites of different generations have been given names. So basically, they asked you a question related to that. Don't confuse yourself. Just learn it as a fact. When you're learning the life cycle, if you want, you can make a side note of what are the names if they ever ask you. But it's not really mentioned in NCERT, I presume. But if they ever ask you, you know the answer, right? Okay, so I guess that was simple. Let's move on. Again, it was a very direct question. If you knew, it was a fact, right? If you knew the answer, the life cycle, and you knew these terms, you could answer the question. Okay, let's move on. Question number 4. The vector for sleeping sickness is, and this appeared in 1989, A. Housefly, B. CC fly, C. Sandfly, and D. Fruit fly. Again, sleeping sickness has come so many times now. So we have a B, CC fly. Okay, that's the vector for sleeping sickness. The answer is B. Okay, so again, direct question that was related to the table that I previously showed, and we already discussed this before. Okay, so we'll move. Okay, so question number 5. The causal organism for African sleeping sicknesses, and then you have your options A, B, C, and D. This appeared in 1989. Now, if you notice, option B is also an African sleeping sickness causal organism, and option D also, because they haven't specified whether it's West Africa or it's East or Central Africa, right? So technically, yeah, they haven't been very specific, but either options could be right, B and D, right? Let's go to the discussion. Yeah, so here West African sleeping sickness is Gambians, East and Central African sleeping sickness is rhodesians. Okay, so it could be either one of the answers unless until they specify which part of the continent is this present. Okay, so let's move on. Okay, so question number 6. Identify the wrong statement with reference to immunity. It's a NEET 2020, very recent question. A. When exposed to antigen, living or dead, antibodies are produced in the host's body. It is called active immunity. Yes. When ready-made antibodies are directly given, it is called passive immunity. Yes, that's correct. So we have to find the wrong statement here. Please make sure you read the question. It says wrong statement. Don't identify the correct statement. Tick it and come back, right? And then you realize that you lost marks because of this. Option C, active immunity is quick and gives full response. No. D, fetus receives some antibodies from mother. It is an example for passive immunity. Yes, that's correct. So D is correct. C is an incorrect statement because the active immunity is divided into two parts. There's a primary and a secondary response. We'll come to that later. So ideally, yes, for question number 6, answer C is the correct answer. But it is an incorrect statement, and the others A, B, and D options are correct. Okay, let's go to the discussion. So active immunity involves the development of B and T lymphocytes. Yes, that's correct. B lymphocytes, so what happens is when they form clones, so see the development of B and T lymphocytes actually acquired immunity. It forms a part of acquired immunity. Now in that, active immunity is when a pathogen, a live or an attenuated virus or a pathogen, bacteria, whatever, enters your system, and these B and T lymphocytes get activated. Okay, they see that antigen does not belong to you, and they know that, so they will get activated and they'll become specific for that particular antigen. Okay, so T lymphocytes will attack it, or B lymphocytes will then eventually form plasma cells, which then form antibodies. Okay, now passive immunity also includes antibodies or T lymphocytes, where antibodies and T lymphocytes are directly given to the patient, okay, from like a donor to a recipient, like that. Okay, so it could be like antibodies are already formed in a lab and they're given, or it could be taken from an animal that has been immunized for that particular antigen, and so they are then extracted and given to a patient. Okay, so that's how this passive immunity is done, but they all fall into the category of acquired immunity. You're acquiring it through your lifetime, right? So B and T lymphocytes create that possibility in your body to acquire immunity against a pathogen. Okay, whether it's bacteria or virus or it's a parasite or whatever the case is. Okay, so B lymphocytes, now what happens is we are specifically talking with respect to the question where it says antibodies. Now, antibodies are formed from plasma cells. And where do these plasma cells come from? They come from the B lymphocytes. Okay, so now B lymphocytes, what happens is they form clones, okay, which can react with an antigen only when they have cell surface receptors that are complementary and recognize a specific antigen. Now, B lymphocytes cannot just randomly, attach anywhere, and also they are specific for a particular antigen. Okay, so they have cell surface receptors that are specific for a particular antigen. Okay, so when they react with an antigen, okay, only when they have cell surface receptors that are complementary and recognize a specific antigen. Now, some B lymphocytes turn into lymphoblasts. Now, when this happens, some B lymphocytes turn to lymphoblasts and then into plasma blasts, okay, which are precursors for plasma cells. So, the process is when an antigen has attached, you have your B lymphocyte here, your particular antigen has attached over here, okay, and this get converted to lymphoblast, and then plasma blasts. Okay, now these plasma blasts divide and produce antibodies which then enter the lymph and the circulatory system. So basically plasma blasts then form plasma cells. The plasma blasts are the precursors for plasma cells, and then they produce antibodies, and these antibodies then enter your lymph and then the circulatory system. Now, passive immunity, okay, this is a different answer. So let's go back to the question first. So when exposed to antigen, okay, antibodies are produced in the host body. It is called active immunity. Okay, so like I said, B lymphocytes are, it specifically says antibodies. That is why I have answered this question related to B lymphocytes, because they are the ones eventually responsible for producing antibodies, right? So only when the B lymphocytes are when an antigen gets attached to a B lymphocyte, which is particular for that, their cell surface receptors on it which are particular for that antigen, they then form lymphoblasts and then plasma blasts, and then the plasma cells, and then which eventually form antibodies. Okay, so yes, to answer this question, when exposed to an antigen, whether it's live or dead, you could even get an infection. Okay, you get an infection just like that, or you could there'll be a dead virus. Vaccines work this way. That's how immunization happens. Okay, so the B lymphocytes then create antibodies that are related to that antigen. Okay, so that's how it works. Now, passive immunity is when antibodies or activated T cells are directly infused into the body. Just understand the difference between active immunity and passive immunity. Okay, and active is when the B lymphocytes have to do the job, or the T or B lymphocytes have to do the job. Okay, they are the ones to produce these antibodies, or T lymphocytes have to act on these antigens, right? But passive immunity, you are directly getting it. You're getting the antibodies directly into your body. You don't have to really do the job. Okay, so passive is when antibodies or activated T cells are directly infused into the body. Now, it can be obtained from the blood from someone else or from an animal that has been actively immunized against a particular antigen, right? Like I previously explained. Now, lactating mothers also pass on IgA antibodies into infants via colostrum during breastfeeding. Okay, so colostrum is the milk that comes out from the breast a few days before parturition and after parturition. So that's a very, very important component of breastfeeding, where the infant needs this colostrum because it is very rich in antibodies. Okay, so now passively the mother is transferring those antibodies into the infant, okay, which helps build the immunity of the infant. Okay. Now, not all lymphoblasts, now we already discussed lymphoblasts, right? Now, not all lymphoblasts formed from B lymphocytes form into plasma cells, okay? They instead form clones of the original B lymphocytes and store themselves in lymphoid tissues and remain dormant until they are activated, okay? So now, like I said, an antigen has, attached to a B lymphocytes, of that particular cell that has a cell surface receptor on it. But not all of them do that. Okay, now you need memory also, right? Because now the B lymphocytes need to remember next time if there's an attack like this, what do we do? Okay, so this is the initial response actually that a B lymphocyte uh, performs or undergoes. Okay, so now not all of them will turn into lymphoblasts and then plasma cells and so on. Some of them get stored. Okay, some of them remember this. Okay, so this particular antigen came in, okay, attached, and now I need to remember this. Okay, so these lymphocytes will remain in the lymphoid tissues. Okay, they will remain dormant there, okay, until they are activated. So they are known as memory B cells. Okay, this is known as primary response. Okay, this is known as primary response. Now, in subsequent action by antigens, the memory B cells will respond faster, making secondary response quicker. Okay, so the initial, now basically the responses also of antigens to a lymphocytes are divided into two parts. Basically, that's what I'm trying to explain here. Okay, so you have a primary response where, for example, you are immunized against a particular virus or a bacteria or whatever it is. Okay, you're immunized. Okay, you are given the first vaccination. The first vaccination you get, and the antigen which is attenuated or it could be inactivated, whatever the case is, it could be a killed bacteria or virus, or it could be attenuated, weakened state. It goes in. Okay, now it goes in, and now it has attached to maybe a particular B lymphocyte, and it has created some response. Okay, it has created response, but it is very slow, and not all of the B lymphocytes will get involved there. Okay, they will then turn into memory B cells and get stored in the lymphoid tissue. So this primary response takes a while. It takes about, let's say, 10 days, okay, for this response to happen. It's called a primary immune response. Okay, and that is why you're given a second dose. Okay, that is why the vaccinations are divided into parts. That is why in immunization schedules you'll have different you'll have for every vaccination, it is designed in such a way that you are divided into different parts. So when you get the second immunization, then it will become faster, because now memory B cells have already been formed against a particular virus or a bacteria, whatever the case is, for that particular antigen. Okay, so now it remembers, oh yeah, okay, so this particular antigen was already there, so now the response will be even faster in producing more lymphocytes against that antigen. Okay, so basically this is what happens. The first primary response is slow. This is important to understand because this is also mentioned in NCRT. The primary response is slow, it is less effective, but the secondary response becomes faster. Why? Because there are memory B cells produced. Okay, there are memory B cells produced. Initially, there were clones of B lymphocytes. Some of them reacted against the particular antigen. Fine, they did their job, or they produced antibodies. But the remaining became memory B cells and remembered that particular antigen. So this is a very important, a part of immunity in our body, and we have this special feature in our body where our cells can remember antigen. So whenever you are attacked again by that particular antigen, your cells remember, the B lymphocytes remember, oh yes, okay, we have been attacked by that same antigen again, and now it is time to produce antibodies. Now the B lymphocytes have already been modified in such a way that they are attracted to that particular antigen. Okay, that is why the secondary response is quicker. Now primary response has a 1 week delay. Okay, this weak potency. Okay, it's not very powerful, it's weak, and it has a short life. Secondary response has a faster response and higher potency and duration. Okay, so this is the difference between primary response and secondary response and immunity. And also please understand the difference between passive immunity and active immunity also. Okay. So yes, antibody IgG can move from mother to fetus via the placenta. So let's go back to the question. I think we'll be able to understand this better. So yeah, when ready-made antibodies are directly given, it is called passive immunity. Okay. So, like I said, obviously in this case it's the correct statement. It's not a wrong statement. So it doesn't fit the answer. But yeah, like I also previously mentioned, that a lactating mother will also transfer IgA antibodies. Also, there's another antibody called IgG. Okay, that's fetus. That's point B. Option D. I'm sorry, option D here. Fetus receives some antibodies from mother. It is an example for passive immunity. That is actually IgG immunoglobulin. Okay, that antibody can easily pass from mother to fetus via the placenta. Okay, so to provide immunity to the fetus. So there are two antibodies, please remember, that are fall under the category of passive immunity in the way they are being transferred from mother to fetus or infant. Okay, so in the case of lactating mothers, and this could be a possible NEET question. Lactating mothers, it is IgA immunoglobulin, and when the immunoglobulin that is transferred from mother to fetus, it is IgG. Okay, it is G and this is A for lactating mothers. Just remember this. Okay, this is very important. All right. Now here active immunity is quick and gives full response. Initially no. Okay, initially no, like I've already said. Okay, so when you are given, of course there is a response, but initially it is not. In the subsequent reaction, yes, it will be faster. Okay, so activity is quick and gives full response, not in the primary response stage. Later on, yes, it does. Okay, let's move on. This was a good question, yeah. So this is a smiley face. All right, let's move on. Okay, question number 7. Which of the following immune responses is responsible for rejection of kidney graft? Okay, NEET 2019. You have A, cell-mediated immune response, B, autoimmune response, C, humoral immune response, and D, inflammatory immune response. So what kind of immune response would you get if there is if it is responsible for rejection of kidney graft? So this particular thing happens in the case of transplantation of organs or tissues. Okay, so in this case it's a kidney graft. Okay, so whenever there's a kidney graft transplantation or any kind of transplantation, obviously there are some cell surface receptors, okay, called HLA, okay, human leukocyte antigen, which are checked. And we'll come to that in the discussion. But to answer this question, what it would be A, okay, cell mediated immune response. Okay, that is the one that's causing the immune response whenever there's a transplanted graft tissue or an organ. Okay, so sorry, sometimes my mouse goes crazy. Yeah. So in a kidney graft transplant, HLA typing is done. Now I already mentioned HLA, it's human leukocyte antigen. Okay, short is HLA. Okay, is done. Now these are actually cell surface receptors. Okay, they're present on our cell. Okay, they're present on our cell. Okay, so in a kidney graft transplant, usually when they are checking for donor and recipient, right, usually you don't get the donor. You must have seen that in movies also, right, where you see that, there's a patient who's very serious and they need a kidney transfer, but you can't get the donor for that, and this whole drama happens in a movie, but this is actually real life. So you don't always get a particular donor which will fit with the recipient, with the patient that needs the kidney transplant, right? So what they do is, the doctors, they do an HLA typing. Okay, so basically they check whether the HLAs of the donor and the recipient match. Okay, so they are cell surface receptors. Okay, so the donor and the recipient should have a close match. Usually you find a close match between twins, or between parent and child. You usually find that in families. Okay, so between the donor and recipient, the closer the match, the higher the acceptance of the graft. So they're like self, antigens. Okay, so basically uh, they kind of recognize that, okay, this person is ours. He or she belongs to, the cell belongs to us. Okay, so the closer the match is, the more the less distance there will be between a donor and a recipient, right? Just imagine if an unknown person enters your house, and they'll be like, no, this person does not belong here, okay, because he's not one of us, right? So but if you know someone, a relative that comes in, or a friend that you know for a very long time comes in, okay, yeah, I know him, he's part of us, so similarly, it's a similar concept that I'm trying to explain here. If you just put a random donor into a recipient, there will be a very severe reaction. Okay, so you need somebody, you need HLAs that are very close to the recipient for the acceptance of that organ or the tissue. So basically now there are 6 HLA present on the tissue cell membrane, and a total of 150 different HLA resulting in various combinations. So there are like 6 that are there on a cell, and there are 150 different types. So just imagine the kind of a number of combinations that are there. So that is why it's very difficult to find a donor and a recipient, a matching donor and recipient, right? It is very, very difficult sometimes to find that. Okay. Now, when tissue rejection occurs, now usually tissue rejection does mostly occur when a donor is, the graft has been transferred from donor to the recipient. There will be obviously some immune response all the time. Okay, so when an immune response happens, now basically that's the answer to our question. So when an immune response happens, which kind of immune response is seen? So it is cell mediated immune response. Okay, so it is the T cells that are responsible for killing the grafted tissue, and their suppression is important for successful transplantation, obviously. Okay, so T cell activation means cell mediated immune response. Okay, so T cells wake up and say, no, this is not one of us, he's an outsider. Okay, this does not match here, it does not belong to us. So they will begin to attack. Okay, so the immunology world, or the immune system, is a different world altogether, like armies of cells, T cells and B cells, that are always fighting to protect us. And they're along with that, they're macrophages, they're neutrophils, the eosinophils, the basophils, all of them are all working together to protect us. Okay, so in this case, of course, immunosuppressant drugs are given by doctors that will help suppress the immune system to allow that graft to survive there. Otherwise, what happens? T cells will start attacking the tissues, and then the tissue cells will die, and then there's no point. Then the organ or the tissue is dead, right? Then there is no point of the transplantation. So there are drugs made which suppress the immune system to allow for the organ or the tissue to survive there in the body. Okay, so to answer our question, yes, T cells are responsible for killing the graft. So which of the following immune response is responsible for rejection of kidney graft? It's cell mediated immune response. Okay, so this is the answer. All right, clear? This was an interesting topic to discuss, transplantation. Okay, so question number 8. Colostrum, the yellowish fluid secreted by mother during the initial days of lactation, is very essential to impart immunity to the newborn infants because it contains, okay, you have A, B, C, and D. We just discussed this, right? This is NEET 2019, again another recent question. So colostrum contains immunoglobulin A, IgA. Okay, that's a short form. They write IgA, they're talking about immunoglobulin A. Okay, and the answer is A. Okay, again, a factual based question. Please remember this. Okay, it's IgA. All right, let's move on then. Okay, question number 9. Which of the following is not an autoimmune disease? It's appeared in NEET 2018. A, psoriasis, B, rheumatoid arthritis, C, Alzheimer's disease, and D, vitiligo. So this is 2018 NEET. Obviously, there's not much written about autoimmunity in NCERT, and they've provided this question. That means you are supposed to know the different diseases, and decide which is not an autoimmune disease, right? So lots of autoimmune diseases in nature, but you need to know some of them. All right, so over here, psoriasis is an autoimmune disease, so that's incorrect. Rheumatoid arthritis is an autoimmune disease. Alzheimer's disease, yes, it's not an autoimmune disease, neurological disorder. And vitiligo, yes, it is an autoimmune disease. So the answer is C. Okay, Alzheimer's disease and neurological disorder. Okay, let's go to the discussion. We will briefly discuss these diseases. Okay, now what is autoimmunity? Okay, now when people lose immune tolerance of their own tissues, it leads to destruction of it. Basically, that's what it is. It comes from the word itself. Even if you didn't know, you just see the autoimmune disease. Okay, that means when the cells begin to attack their own cells. Okay, T cells, B cells begin to attack their own cells. Okay, that's autoimmunity. Okay, so that is not a very good condition to be in, because you don't want your cells to your immune cells to attack your own cells, right? So but it comes from the term autoimmune disease. You can see, you can understand it from the word itself. So when people lose their immune tolerance, okay, when the T cells lose their immune tolerance, they begin to attack their own tissues. Okay, leads to destruction of it. So it usually occurs when there's destruction of tissues, leading to increased circulation of self-antigens in the system, and hence activating acquired T-cell immunity or antibodies. So usually it occurs, not all the time. Okay, please, this is only one of the conditions, is where there's destruction of tissues leading to increased circulation of self-antigens that are present. This is one particular condition which it can occur, and hence they activate the T cells and the antibodies. When there are too many self-antigens circulating around in your body, and remember I told you what is self-antigen, non-self-antigens happens in transplantation also. Self-antigens are part of us. Okay, now but there could be an instant where these immune system is not working like it should, right? It usually shouldn't attack your self antigens. That's not what it should be, but it tends to lose its immune tolerance and then begins to attack. Okay, these T cells and antibodies become activated and they begin to attack their own cells. Okay, so that's autoimmune. Now there are various autoimmune diseases. The first option that we saw was psoriasis, right? So it is a dermatological disorder. It is a chronic inflammatory skin disorder. So what happens, it is characterized by erythematous and demarcated papules and rounded plaques. Just giving you a short brief on what psoriasis is. If you want, you can even Google and check what it looks like, so you get an idea what I'm talking about. It's a skin disorder. So it is known to activate T cells, which enhance cytokines response for keratinocyte hyperproliferation. Okay, so these T cells, then they enhance these cytokine responses, okay, which then cause hyperproliferation of keratinocytes. Okay, they are epidermal cells, okay, present on the skin. Okay, so these T cells then tend to cause too much of proliferation or division of epidermal cells. Okay, that's psoriasis. You usually find, like, a reddish skin disorder on the body, on the skin, and like I said, it's erythematous, and it's a chronic inflammatory skin disorder. It's erythematous and it has papules and rounded plaques. Okay, so unfortunately I can't put a photo of that here. I don't know if YouTube will allow me to do that, but what you can do is do a simple search on Google if you're brave enough. Yeah, of course, you're gonna be future doctor, so you need to be brave enough to see that. But yeah, you get an idea of what I'm talking about, what psoriasis looks like. Okay, so now rheumatoid arthritis is again a chronic inflammatory arthritis disease and cause articular cartilage and bone destruction. Okay, the T cells are self-reactive and cause chronic inflammation. Again, the T cells are attacking its own cells. Okay, so that it causes rheumatoid arthritis. Okay. Vitiligo is a condition in which there is loss of pigment from areas of skin, forming white patches. I'm sure maybe you have seen some people who have melanin. They don't have melanin in certain patches of the skin. They're like whitish in color. Okay, compared to their normal skin color, which has melanin in it. Okay, so that's called vitiligo. It's an autoimmune disorder. So it's an autoimmune disease, and it is associated with other autoimmune diseases such as Hashimoto's disease, Graves' disease, pernicious anemia, Addison's disease, alopecia. So those who have vitiligo have also have certain other autoimmune diseases also. So these are some of them. Thyroid glands are frequently associated, occurring in up to 30% of the patients with vitiligo. So yeah, basically that's what happens. Thyroid glands get affected. Okay, with patients in patients with vitiligo. Now, Alzheimer's disease, I'm sure you're aware of this. We discusses neural control and coordination. Alzheimer's disease is the common cause of dementia in elderly people, and the clinical features are these three. So it's amnesic type of memory impairment, the deterioration of language, and this visuospatial defects. Okay, so B amyloid plaques are formed in hippocampus, temporal cortex, nucleus basalis, and locus ceruleus parts. They're all parts of the brain. Don't worry about all this. Basically, this is what happens in Alzheimer's disease. It's a neurological disorder. It has nothing to do with autoimmunity. Okay, they're parts of the brain resulting in disruption of cholinergic and norepinephrine nerves. Okay, so basically the production of acetylcholine gets affected, okay, because of this. All right. Okay, so basically I've just briefly discussed what are those diseases and how they are autoimmune, and so that you know what I'm talking about and how to answer this question. Obviously you could have used the process of elimination if you knew Alzheimer's disease is not an autoimmune disease, it's a neurological disorder. That would be easy to answer the question there. That was a trick there. Okay, you are not really expected to know maybe all the autoimmune diseases that are there. There's a whole list of autoimmune diseases, but if you knew that Alzheimer's disease is a neurological disorder, the question directly asked that, which is not an autoimmune disease. You could have easily answered this question. Okay, let's move on then. Yeah, it's a neurological disease and not an autoimmune disease. That's about it. Let's move on. Okay, so question number 10. Transplantation of tissues or organs fails often due to non-acceptance by the patient's body. Which type of immune response is responsible for such rejections? NEET 2017, we just discussed transplantation. So which type of immune response is responsible for such rejection? You should be able to answer it quite easily. It's cell-mediated immune response. Okay, the T cells begin to attack. So answer is A. So there's no HLA match between the patient and donor, then the transplant graft will be rejected. Almost all the time there is some immune response, even if there's HLA typing properly done and checked. This will result in activation of T cells, which then attack the transplanted graft or organ. Okay, so I already discussed HLA typing and transplantation in the previous question. So you go back and watch that discussion again if you need to. So T cells are cell-mediated immune response. Please remember that. B cells are humoral mediated immune response. So yeah, remember these differences between T cells and B cells. Okay, so question number 11. MALT, M-A-L-T, constitutes not the malt as in malt, the other one. MALT, it's an acronym, constitutes about something percent of the lymphoid tissue in human body. Okay, appear in NEET 2017. A 20%, B 70%, C 10%, and D 50%. Okay, so MALT, M-A-L-T, constitutes about 50% of the lymphoid tissue in the human body. Okay, so the answer is D. MALT, or mucosa associated lymphoid tissue, that's the full form for MALT. Okay, constitutes 50% of the lymphoid tissue. Now, what is this MALT? I'm sure you, if I explain it to you, you'll understand. Okay, so this is MALT. Okay, so these are aggregations that are seen in mucosa of alimentary, respiratory, and urogenital tract. Okay, so this is what these mucosa-associated lymphoid tissues are. Now, MALT structure consists of lymphatic follicles. Okay, so I'm just briefly explaining what MALT, the lymphoid tissues look like. Okay, the mucosa-associated lymphoid tissue. Okay, so they have a nodule and they have this parafollicular zone. Okay, so the lymphatic follicles, which are also known as nodules, which contain B lymphocytes, and diffuse lymphoid tissue, which are the parafollicular zone present in between lymph follicles, contain T lymphocytes. Okay, so the main part contains the nodule, which has B lymphocytes in it, and the parafollicular zone, which present between the lymph follicles, they contain the T lymphocytes. Okay, they're diffused lymphoid tissue. The aggregates lie close to the lining of the epithelium and they are not surrounded by a capsule. Okay, so lymph nodes usually are surrounded by a capsule, but this MALT are not surrounded by capsules. Okay, and they have a supporting network of reticular fibers. They only produce lymphocytes and pass it into lymph nodules through the efferent lymphatic vessels. Okay, so that is how they leave. Okay, they only produce the lymphocytes and they pass it into the lymph nodes through the efferent lymphatic vessels. Oh, sorry, went to the next question. To answer your question, yes, it constitutes 50% of the lymphoid tissue. Okay, and they are present in the alimentary, respiratory, and urogenital tract. Okay. And these aggregation are I have written here mucosa of alimentary. That means, for example, Peyer's patches. Okay, they are mucosa associated lymphoid tissue. Okay, tonsils that are present, they are also mucosa associated lymphatic tissues, lymphoid tissues. Okay, so these are some of the examples of MALT and they constitute 50% of the lymphoid tissue. Okay, question number 12. Antivenom injection contains preformed antibodies while polio drops that are administered into the body contain what? Okay, this appeared in NEET 2016. A. Gamma globulin, B. Attenuated pathogens, C. Activated pathogens, and D. Harvested antibodies. Okay, so polio drops are oral polio vaccine. Okay, and they contain attenuated pathogens, not activated pathogens. Answer is B. Yeah. Gamma globulin, no, they don't contain antibodies, and harvested antibodies, no, they don't contain that too. It has to be B. Okay. So the oral polio vaccine consists of attenuated virus strains. Okay. We've already talked about Salk and Sabin polio vaccines, right? So this is different. This is oral polio vaccine now that they use to give infants. They constitute of attenuated viral strains. Okay, please remember this. This is a good question. I mean, obviously it's factual based, but you could have easily answered it if you had carefully checked all the options available. But just remember it. Okay, it contains attenuated viral strains. Okay, let's move on. Question number 13. In higher vertebrates, the immune system can distinguish self cells and non-self. Yes. If this property is lost due to genetic abnormality and attacks self cells, then it leads to, we already discussed this, right? So NEET, it's appeared in NEET 2016. So A, autoimmune disease, B, active immunity, C, allergic response, and D, graft rejection. So when the immune system begins to attack our own cells, it means it is autoimmune disease. Okay, it leads to autoimmune disease. Answer is A. We discuss this. When people lose immune tolerance of their own tissue, it leads to destruction of it. Usually occurs when there is destruction of these tissues, leading to increased circulation of self antigen. That's one of the ways by which these cells, B and T lymphocyte, begin to lose their immune tolerance. Okay, clear? This is known as autoimmune disease. Right, let's move on then. Question number 14. If you suspect major deficiency of antibodies in a person, to which of the following would you look for confirmatory evidence? This appeared in twice in 2007 and 2015. Okay, so if you suspect major deficiency of antibodies in a person, to which of the following would you look for confirmatory advice? Okay. So you have A, hemocytes, B, serum globulins, C, fibrinogen plasma, and D, serum albumins. So what are basically antibodies is what they are trying to test us on. Okay, so what is the basic nature of antibodies? What are they actually? Okay, so we know that they are plasma proteins. Okay, but exactly what? Right? So they are actually globulins. So the answer is B, serum globulins is where you will look for. Okay, if you see a major deficiency of antibodies, do we have gamma globulins or not? So these are antibodies. Okay, we'll come to the discussion in some time. The answer here is B. Okay, so serum contains immunoglobulins known as antibody. Okay, now there are different names. So there's antibody that we know. Okay, now antibodies form a part of plasma proteins. Which kind of plasma protein? They are globulins. Globulin type of plasma protein. So antibodies are known as immunoglobulin. Okay, now which kind of immunoglobulin? They are gamma globulin. So if you see the term gamma globulin also, you should know that they are talking about antibodies. Please don't get confused, right? What is gamma globulin? Gamma globulin means antibodies. Okay, the basic structure of antibodies is what you see on your screen here now. Okay, so they are Y-shaped proteins, and one end binds to the antigen and the other end attaches to the cell that they have suitable receptors on its surface for. Okay, so you see the heavy chain, you see the light chain here, you see this heavy chain here, and these are the antigen binding site. Okay, the antigens get trapped there. Okay, so they are designed in such a way that they can capture these antigens. Okay, and they are these heavy and light chains are connected by disulfide bridges. Okay, disulfide bridges. Now one more thing. Now I said they're plasma protein. They're proteins. We know that, right? They're globulins. But what kind of protein are they? So if they ever ask you this question, you should remember that they are glycoproteins. Okay, if they ever ask you that in a question, they are actually glycoproteins. Okay, so to answer your question, yes, you will look at serum globulins to confirm whether there is a deficiency of antibody or not. So that should be your evidence. Okay, the others clearly don't match. A, C, and D don't match. Let's move on then. All right, question 15. Which of the following immunoglobulins does constitute the largest percentage in human milk? This appeared in 2015. We discussed this. Also, it is IgA. So IgA is present in large amounts in colostrum in breast milk. So the answer is A. Okay, I've already discussed this. So it forms a part of passive immunity where the mother transfers this immunoglobulin to the infant. Okay, so it's IgA antibodies. Okay, next question. Question 16. Grafted kidney may be rejected in a patient due to, and we already discussed this twice. This appeared in 2015. You have A, passive immune response, B, innate immune response, C, humoral immune response, and D, cell-mediated immune response. When a grafted kidney may be rejected in a patient, then cell-mediated immune response, the T cells get activated and begin to attack the grafted tissue. So the answer is D. Okay, may be rejected due to cell-mediated immune response. Okay, so let's move on. Clear, right? Question 17. Increased asthmatic attack in certain seasons are related to, this appeared in 2007. A, eating fruits preserved in tin containers. Inhalation of seasonal pollen, yes. Low temperature, no. Hot and humid temperature, no. So the answer is B. Okay, let's do the discussion. So when allergens are inhaled, they attach to IgE antibodies. So people that have large amount of IgE antibodies are generally prone to this asthmatic attack. Okay, so now how does this work? Now when allergens like seasonal pollen or any kind of allergen a particular person is allergic to, okay, now the allergen behaves like an antigen, okay, and the person inhales, let's say, pollen grains, okay, and the person is allergic to pollen grains. Okay, now they inhale and they attach to the IgE antibodies. Okay, now these antibodies are called reagin. Okay, an allergen reagin reaction takes place. Okay, so this reaction takes place. Now IgA binds also with basophils and mast cells, as well as, so when IgE binds with both the allergens as well as the above mentioned cells, then an immediate change occurs in the membranes of mast cells and basophils. So now allergen reagin reaction has happened between the pollen and the antibody. Now this antibody is also attached to basophil and mast cells. Now this whole reaction between pollen and this antibody, and let's say it has attached to mast cell, okay, and this is pollen grain and this is your IgE antibody. Okay, now this causes a reaction what, in this cell membrane of these mast cells. Okay, now the mast cells and basophils rupture and release chemical agents such as histamine, protein, or the series of uh, chemical substances that are released. Okay, so when mast cells or basophils rupture, series of chemical are released called histamine, protease, leukotrienes, eosinophil chemotactic substance, neutrophil chemotactic substance, heparin, platelet activating factors. You know, that chemotactic substance of eosinophil and neutrophil means they attract eosinophils and neutrophil towards them. Okay, it's an ability. There are certain substances which attract eosinophil, that's eosinophil chemotactic substance, and neutrophil chemotactic substances when neutrophils are attracted to them. So they're calling eosinophils and neutrophils to come save us. Basically that. This heparin, this platelet activating factors, so basically all these factors are chemical factors are released. Okay, so now in the case of asthma, the reaction takes place in the bronchioles of the lungs. Okay, and leukotrienes, which are slow reacting substance of anaphylaxis, which causes spasm of bronchiolar smooth muscles. Okay, so in the bronchioles, let's say this reaction has happened. Okay, the person has inhaled this pollen grains, IgE antibodies, mast cells, it has attached to that, the mast cells have ruptured and it has released. Now basically in the case of asthma, it is found that leukotrienes are released. Now, leukotrienes are really slow reacting substances of anaphylaxis, which is again a type of allergic reaction, but we'll not go into that, just understand this asthma. Okay, now when these leukotrienes are released, what happens is it causes smooth muscle spasm. Okay, and the contraction of bronchioles begins to happen and the person finds it difficult to breathe. It finds it very difficult to breathe, and so the person will only come back to normal when those substances are removed from the body. Okay, those reactive substances are removed from the body, so the person has difficulty in breathing until the products of the allergic reactions have been removed. Okay, usually it is said that antihistamines also don't work in the case of asthma attack, unless the person obviously has difficulty in breathing, and only when the products of the allergic reactions are removed, does the person feel relieved. Okay, so what I've done initially is explain what an allergic reaction is. Okay, so in the case of IgE antibodies, obviously there is one more type of allergic reaction where there's delayed T cell response, but we'll not go into that. Don't confuse yourself too much with all this. Just understand, sorry, that IgE antibodies are responsible for allergic reaction. In this case, there's an allergen such as pollen grains, or it could be any other substance that a particular person is allergic to, and they then they attach the IgE antibodies that then attach to the mast cells or the basophils, and this whole three reaction over here results in cell membrane rupture of the mast cells, okay, which release a whole series of chemical factors. Okay, now in the case of asthma, this thing happens in the bronchioles, okay, in the respiratory system. Okay, now when this happens, there is a bronchial smooth muscle spasm. Okay, so they begin to contract. Okay, the person finds it difficult to breathe. Okay, it finds it difficult to breathe unless and until that those reactive products are removed, okay, from the system. Okay, so this is what asthma attack is. Okay, when they talk about asthma attack, this is basically what's happening. Okay, there's a reaction that has happened in the body and the person is allergic to the particular allergen in nature, and so these reactions happen inside the body. Okay, so if we go back to our question, increased asthmatic attack in certain seasons, like spring season, you'll find a lot of pollen grains in the air. Okay, and there are people who are allergic to that pollen grains, and so that results in a lot of asthmatic attacks because of that. All right, clear? Let's move on. Okay, question number 18. Lysozyme that is present perspiration, saliva, and tears destroys, submitted in 2007. You have certain types of bacteria, all viruses, most virus infected cells, certain fungi. So lysozyme is responsible for destroying certain types of bacteria, right? They're antibacterial. So the answer is A. So yeah, lysozyme is an enzyme that is secreted by the submucosal gland, neutrophils, macrophages, and salivary glands. They're secreted by the zymogen cells in deeper ends of the intestinal crypts. The eosinophilic granules of the fibrous tissue of the respiratory mucosa also secrete lysozyme. Okay, so you find lysozyme in these various parts of the body. Okay, now they damage or kill bacteria by either lysing the cell wall, they're disrupting the bacterial membranes, and by activating autolytic activity from the bacterial cell wall. So the bacteria kills itself. Okay, so it's autolytic activity from the bacterial cell, a certain substance which will kill the bacteria itself. Okay, they present the bacterial cell wall. They disrupt the bacterial membranes, they lyse the cell wall. So these are different ways by which lysozyme is very effective in killing bacteria. So they are antibacterial. It was a very simple question, right, to answer. Just remember, lysozyme is antibacterial. That's all. Okay, so I guess we can move on. Question number 19. Antibodies in a body are complex. So in 2006, A. Glycoproteins, B. Lipoproteins, C. Steroids, D. Prostaglandins. So we know they're proteins, but they're definitely not lipoproteins. They are glycoproteins. We discussed that, right? So the answer is A. So antibodies are glycoproteins. They contain protein part that's two heavy chains and two light chains, and they also have a carbohydrate part also. That is why they called as glycoproteins. If you have to be very specific, like in this question, they ask you what kind of protein is antibody. We know it's a protein, but what kind of protein? So it's a glycoprotein, because it has a carbohydrate part also. Okay. Okay, all right. Let's move on. Question 20. Damage to thymus in child may lead to, somewhere in 2005, A reduction in hemoglobin content of blood. No. A is incorrect. B, a reduction in stem cell production. No. Loss of antibody mediated immunity. No. Loss of cell mediated immunity. Yes. Answer is D. If the thymus is absent or damaged or whatever the case is, T cell production will be affected. So thymus is responsible for pre-processing of T cells. Okay, so before the T cells are actually differentiated and stored in the lymph nodes, there is a pre-processing stage which happens in the thymus. Okay, thymus is very, very important. That's how the T cell part comes. The letter T comes from thymus actually, and it's an easy way for you to remember. And thymus is responsible for this. Okay, before they are stored in the lymphoid tissues, the lymph nodes, spleen, tonsils, and intestine. So hence, if thymus was damaged or absent or whatever the case is, then there will be loss of cell-mediated immunity. Okay, so thymus plays a very, very important role in cell-mediated immunity. Okay, good question. You had to understand what the role of thymus is, okay, in the immunity. And let's move on then. Question 21. Short-lived immunity acquired from mother to fetus across placenta or through mother's milk to the infant is characterized as, we discussed this before, right? This appeared in 2003. You have active immunity, passive immunity, cellular immunity, innate, non-specific immunity. No, none of them are incorrect except for B. Okay, passive immunity is what's responsible when the antibody is just handed over to you. It's passive. You're not really doing the job of creating these antibodies or activating T cells, right? So in this case, we are talking about antibodies. So the mother is basically handing over the antibodies to the infant or to the fetus when she's pregnant. Okay, so this is called passive immunity. Answer is B. So passive immunity is when the antibodies are directly injected or passed on to the other person. So IgG antibody can easily pass through the placenta to the fetus. IgA is present in the colostrum, which is imparted to the infant by the mother at the time of breastfeeding. We've already discussed this. IgG and IgA. Remember these names. If they ever ask you in the question in the exam, you know the answer, right? Okay. So this was simple. Okay, so we've come to the end of this video lecture, and I hope you enjoyed today's video lecture. I hope you've learned something new today. And don't forget to like, share, and subscribe to MG NEET Biology, and I'll see you in the next video lecture, right? So, bye.